Welcome back to the University of Melbourne Biology series. This is episode 4, and today we ask two of the biggest questions in cell biology. Where did the complex eukaryotic cell, the kind of cell that builds you, come from? And once such a cell exists, how does it make another one of itself? These two stories are deeply connected. To divide reliably, a cell first needs the very machinery whose origins we are about to trace. A nucleus to hold its instructions, organelles to power it, and a scaffold to pull itself apart. Over the next 12 minutes we will follow that thread from the origin of the eukaryotic cell all the way to the moment it splits in two. Let us begin at the beginning. The feature that defines a eukaryotic cell is its nucleus, so let us start there. The nucleus is a double membrane-bound organelle, and inside it sits the cell's DNA, wound around proteins called histones and organized into chromosomes. Studded through that double membrane are nuclear pores, channels that control which molecules move in and out. The nucleus is the site of two essential jobs: it is where DNA is replicated, and it is where genes are transcribed. And it does not stand alone in the cell, its membrane is continuous with the endoplasmic reticulum, so it is wired directly into the cell's internal membrane system. This is the control center, and remembering what lives inside it will matter when the cell comes to divide. But where did such an elaborate structure come from? The leading idea is surprisingly homemade. The nucleus likely evolved from the invagination of the plasma membrane, the outer membrane of an ancient prokaryote folded inward, wrapping itself around the nucleoid, the loose region of DNA, until that DNA was enclosed in its own membrane compartment. There is a neat piece of evidence baked into the cell for this. Recall that the nuclear membrane is continuous with the endoplasmic reticulum. That continuity is exactly what you would expect if the nuclear envelope was originally part of the cell's own membrane system, pinched inward rather than imported from outside. The nucleus, in other words, was an inside job. But the next two organelles tell a very different origin story. Consider the mitochondria, the organelles responsible for cellular respiration, the cell's power supply. Like the nucleus, a mitochondrion is a double membrane-bound organelle, but its history is not homemade at all. Mitochondria likely originated from primary endosymbiosis, an ancient host cell engulfed a free-living purple bacterium, and instead of digesting it, kept it alive inside. Over evolutionary time that captured bacterium became a permanent resident. The strongest clue is that mitochondria are semi-autonomous. They still carry their own DNA and their own protein synthesis machinery, a private genome and ribosomes that no other organelle in the cell possesses. That is exactly what you would expect from a former bacterium that never fully gave up its independence. The cell did not build its power plant, it captured one. Plant and algal cells pulled off the same trick a second time, with a different partner. Chloroplasts are the double membrane-bound organelles responsible for photosynthesis, and they too likely originated from primary endosymbiosis, this time of a photosynthetic cyanobacterium engulfed and retained by a host cell. So the same evolutionary move, capture and keep, gave the cell both its respiration and its photosynthesis. But chloroplasts have an extra chapter. Some of them evolved through secondary endosymbiosis, in which a eukaryotic cell engulfed another eukaryotic cell that already contained a chloroplast. That is endosymbiosis nested inside endosymbiosis, a cell within a cell within a cell. The eukaryotic cell, then, is not a single invention but an assembly of captured and refolded parts. Now that we have built one, let us watch it reproduce. A eukaryotic cell does not divide on a whim. It follows an ordered program called the cell cycle. That cycle has two broad parts, interphase and mitosis. Interphase is by far the longest stretch, accounting for about 90% of the whole cycle, and it is itself divided into three phases in sequence, G1, then S, then G2. Mitosis, the much briefer part, is the process of nuclear division. So a cell spends most of its life not dividing at all, but in interphase, growing and preparing. The headline to hold on to is this: the dramatic moment of splitting is short, while the careful preparation that makes it safe takes up almost the entire cycle. And one step in that preparation is the most important of all. Before a cell can split its contents in two, it must first make a complete copy of its DNA, and that copying is DNA replication. Replication has one defining feature: it is semi-conservative. That means each new DNA molecule is built from one strand of the original molecule paired with one freshly synthesized strand. The original is never thrown away, it is preserved as a template inside every copy. The building itself is directional. Nucleotides are added in one direction only, from the 5' end toward the 3' end, and the enzyme that carries this out is DNA polymerase III. So replication is faithful by design: keep half of the old, build half that is new, and you guarantee each daughter cell inherits an accurate copy of the instructions. With the DNA copied, what stops a cell from rushing ahead before it is ready? The cell cycle is tightly regulated, and the regulators are two families of molecules working as a pair: cyclins and cyclin-dependent kinases, usually shortened to CDKs. Together, cyclins and CDKs control progression through the cell cycle, governing whether the cell advances from one phase to the next. Think of them as the cell's gatekeepers, the molecular switches that decide when interphase gives way to division. This control is not a luxury. Because each daughter cell must receive a correct and complete copy of the genome, the cell cannot afford to move forward at the wrong moment, and cyclins and CDKs are what hold that timing in check. Eukaryotes, then, divide through this elaborate, tightly controlled cycle. Prokaryotic cells do it far more simply. A prokaryote divides by binary fission, a process in which the cell splits into two identical daughter cells. There is no nucleus to dismantle and rebuild, and crucially, binary fission does not involve mitosis at all. It is a fundamentally simpler process than the eukaryotic version. The contrast is instructive. Mitosis is a complex nuclear division, an intricate choreography needed precisely because eukaryotes package their DNA inside a nucleus and across multiple chromosomes. The prokaryote, lacking that complexity, can take the direct route: copy, split, done. Same end goal, two daughter cells, reached by two very different paths. Let us put the two side by side so the comparison is clear. Both processes end in the same place: a cell becomes two. But the route differs sharply. Binary fission is the prokaryotic route, splitting the cell directly into two identical daughter cells without any mitosis. Mitosis is the eukaryotic route, a complex nuclear division in which the nucleus itself is divided. The reason for the difference traces straight back to where this episode began. Eukaryotes carry their DNA inside that membrane-bound nucleus we built earlier, so dividing it demands an elaborate, regulated procedure. Prokaryotes have no such nucleus, so they need no such procedure. Structure dictates strategy. How a cell is organized determines how it must divide. There is one more player without which none of this division could happen: the cytoskeleton. The cytoskeleton is a network of protein filaments running through the cell that provide structural support, maintain cell shape, and drive cell movement. It is built from three kinds of filament, each with its own job. Microfilaments are involved in cell contraction and movement. Intermediate filaments provide mechanical strength and stability, holding the cell together under stress. And microtubules are involved in cell division and in intracellular transport, moving cargo around inside the cell. That third role is the one to mark. The microtubules that shuttle cargo are the same family of filaments the cell calls on to physically separate its contents when it divides. Step back and watch the whole picture connect. The eukaryotic cell was assembled from captured and refolded parts, a nucleus formed by membrane folding inward, and mitochondria and chloroplasts taken in whole from free-living bacteria. Holding all of it together is the cytoskeleton, the protein scaffold that gives the cell its shape. When the time comes to reproduce, that cell runs an ordered cycle, spends most of it in interphase faithfully copying its DNA strand by strand, and lets cyclins and cyclin-dependent kinases decide when to proceed, before mitosis divides the nucleus and the cell becomes two. The prokaryote reaches the same destination by the far simpler road of binary fission. From the origin of the eukaryotic cell to the instant it splits, every piece we have met today is part of one continuous story. See you next time.